Okay, let's unpack this. You've sent us some really incredible source material. It's all about this constant battle, this molecular arms race between bacteria and viruses, the phages.
Yeah, it's amazing stuff. And the defenses bacteria are just, well, mind-blowing sometimes.
Really. And, many of us have heard of CRISPR-Cas9, right? That bacterial system we've actually repurposed for gene editing. It's even FDA approved now. A prime example of bacteria fighting back.
It really is. It highlights the sheer ingenuity happening at this tiny, tiny scale. Yeah. Bacteria are constantly under siege, so they've evolved these really sophisticated defense layers. CRISPR gets the headlines, sure, but it's definitely not the only game in town.
Exactly. Not the only defense, and maybe not even the only trick up CRISPR's sleeve either.
Right.
And that's what this deep dive is about. We're going a bit deeper, focusing on this other group of bacterial immune proteins. They're called CARF effectors. And your sources really spotlight some recent research, mainly from Luciano Marafini's lab at Rockefeller and Dinshaw Patel's group over at Memorial Sloan Kettering. They've been pioneers in figuring out these, let's say, less famous defense systems.
Yeah, their teams are really digging into how these CRF effectors work. Often the strategy involves, well, sacrificing the one infected cell to save the whole bacterial community. A bit dramatic, but effective.
Yeah. Cell sacrifice, yeah. And the star player in this particular deep dive, based on their latest study in Science, is this newly found CAR-IRF effector. They've named it CAT1.
CAT1, right?
So our mission here, looking at the paper by Christian Baca, Pooja Majumdar, James Hickling, Patel, and Marafini, is to really get into the nitty-gritty of that study. Yeah. We want to figure out what makes CAT1 tick, why it's so effective, and frankly, why it's so surprising.
It is quite surprising, yeah.
We'll focus on its unique strategy and its structure, which turns out to be unexpectedly complex.
It's a fantastic illustration of how much we still don't know, even about processes happening right under our noses, metaphorically speaking, in the microbial world.
Okay, so before we zero in on CAT1 itself, maybe let's quickly revisit the landscape of bacterial defense. Just set the stage. We mentioned CRISPR. The study reminds us it's adaptive immunity, right? Learns from past infections, protects against phages. The classic view is CRISPR RNA finds the foreign code.
The viral code, yeah.
And then it signals a Cas enzyme like Cas9 to come in and just snip, cut the viral stuff.
That's the textbook picture, the sort of genetic scissors idea. But what the field, and especially Marafini's lab, has been uncovering is that CRISPR systems are way more versatile. They do more than just cut things.
Our tools in the toolkit.
Exactly. And they've been particularly focused on these CARF effectors, which you find in certain CRISPR systems, like CRISPR-Cas10. These get switched on when a phage attacks.
Okay, so before CAT1, what did they think these CARF effectors generally did?
Well, the thinking was, and they had examples, that CRF effectors worked by making the inside of the infected cell basically uninhabitable for the virus, just wrecking the place, essentially.
Wrecking the place. I like that. The source mentions a couple of examples like CAM1. It messes with the cell membrane's electrical charge, depolarizes it.
Yeah, shorts it out, basically. Or another one, CAD1, which triggers what the researchers called molecular fumigation.
Molecular fumigation.
Yeah.
Sounds intense.
It does, like flooding the cell with toxic stuff.
Yeah.
But the end goal for both is the same. Stop the virus from making copies of itself. Stop it from spreading. Protect the colony.
Right. Different tactics, fumigation versus electrical chaos, but the same population level goal. Okay, so that brings us to CAT1. If they already knew CRF effectors did these kinds of disruptive things, how did they even spot CAT1, which, as you said, has a different approach?
Well, finding CAT1, according to this study, involved using a tool called FoldSeq. It's a structural search tool. Right. They were basically looking for proteins that had a similar shape to known CIRF effectors. The hypothesis was similar shape might mean related function, even if the specifics were different.
So looking for family resemblances in shape. And they found CAT1. And its strategy wasn't depolarization or fumigation. It was something else entirely.
Completely different. CAT1 goes after something incredibly fundamental, a molecule called NAD plus BIC.
NAD plus IC. Okay, I know that's important. It's like cellular energy currency.
Exactly. It's absolutely essential. Cells need it for, well, almost everything. Making energy, repairing DNA, signaling. It's critical. CAT1's main job is to find NAD plus and just cleave it. Chop it up.
Wow, okay. So it destroys the cell's money, essentially.
Yeah.
But how does CAT1 know when to start wrecking the treasury? It can't just be active all the time, surely.
No, definitely not. It needs a trigger, an alarm signal. And like a lot of these immune proteins, it gets activated when specific messenger molecules show up. For CAT1, the trigger is something called cyclic tetraadenylate, CA4. These CA4 molecules only get produced inside the bacterium when it senses it's being invaded by a virus. So CA4 showing up is the red alert phage attack signal that switches CAT1 on.
Got it. So phage invades cell, makes CA4 binds to CAT1 activates and starts chopping NAD plus CAT1. Okay, what happens then to the poor infected cell?
Well, the study explains that as CAT1 cleaves more and more NAD plus SCAT, the levels drop. And once they drop low enough, the cell just stops. Yeah, enters what they call a growth arrest state. You could picture it like a metabolic freeze. Everything that needs NAD plus just grinds to a halt. Energy production, repairs, everything.
A metabolic freeze. That's a very clear image.
Yeah.
And this freeze presumably achieves the same ultimate goal as CAM1 or CAT1, stopping the virus.
Exactly the same goal. If the cell's machinery is frozen, the phage inside can't use it to replicate. It can't build new virus particles. It can't spread.
So the infection is contained.
Right. The infected cell is sacrificed, essentially put on ice, to protect the rest of the bacterial population. So it's population immunity again, but achieved through this totally different tactic, metabolic shutdown, instead of, blowing up the membrane or releasing toxins.
Okay, so the overall strategy, population immunity via cell sacrifice, fits the URF effector pattern. But the method is novel, this metabolic freeze by NAD plus destruction. But what the source really seems to emphasize as the big surprise, maybe even more than the NAD plus trick, is CAT1's physical structure.
Oh, absolutely. This is where the cryo-EM work, led by Pooja Majumdar, dropped a bit of a bombshell. Cryo-EM lets you see these molecules in incredible near-atomic detail.
Right. High-res molecular snapshots.
And what they saw was that while CAT1's function fits the family, its form, how it actually builds itself upon activation, is, well, it's remarkably complex, and quite unlike other effectors they'd studied.
Okay, break down the structural complexity for us. The study mentions monomers, dimers, things being glued. What's going on there?
Right, so you start with individual CAT1 proteins, the monomers. They naturally pair up, forming dimers. It's pretty standard.
2 units together.
But then when the CA4 signal molecule, the phage alert signal, binds to the dimer, it does something really cool. It doesn't just switch it on. It acts like molecular glue.
Glue?
Yeah, it causes these activated dimers to link up end to end, forming these incredibly long thread-like structures. Filaments.
Whoa, filaments. So inside the cell, this protein starts building long threads when the alarm goes off.
Precisely. And they're not just inert threads. The analysis showed these filaments create specific grooves or pockets along their length. Sticky molecular pockets, as the source puts it.
Sticky pockets? What for?
To trap NAD plus 7. So the filament itself becomes this long scaffold that actively captures the NAD plus molecules, bringing them right to the enzymatic part of CAT1 to be cleaved. It's like a molecular assembly line for destruction.
Okay, that's already pretty sophisticated. Monomers, dimers, activated and glued by CA4 into filaments that trap and chop NAD plus core. But you said it gets even more complex.
It does, yeah. The cryo-EM structures revealed these filaments don't just float around individually, they actually interact with each other. They start twisting together.
Twisting?
Yeah, forming larger bundles. First, they saw them forming trigonal spiral bundles. Think of 3 filaments winding around each other like a 3-stranded rope.
Okay, trigonal bundles, 3 filaments coiled.
Exactly. But wait, there's more.
Oh.
Under certain conditions, or perhaps further along in the process, these trigonal bundles can actually expand and incorporate more filaments, forming pentagonal spiral bundles.
Pentagonal, so now 5 filaments are twisting together.
5 filaments, yeah. It's this hierarchy of assembly. Monomers to dimers, dimers glued into filaments, filaments bundling into 3s, and then potentially expanding into bundles of 5, layers upon layers of self-organization.
Wow, that is unexpectedly intricate for a single bacterial defense protein. Does the study say why it builds these elaborate bundles? What's the point of the 3s and 5s?
That's the million-dollar question, really. The researchers note that the specific biological reason for these higher-order structures, the trigonal and pentagonal bundles, isn't clear yet. It's something that definitely needs more investigation. But it highlights this incredible, almost architectural complexity in a single immune protein, which is pretty unusual. It's not just an enzyme, it's building structures.
Building structures to fight viruses.
Okay.
And speaking of unusual things about CAT1, the study points out something else too. It often seems to work. by itself.
Yeah, that was another interesting finding. Cat1 is part of what's called a type 3 CRISPR system. And normally with type 3 systems, you expect to see at least 2 different kinds of enzymatic activity working together to get the full immune effect.
Okay, like a team effort usually.
Right. Maybe one part targets the phage's genetic material, the RNA, and another part does something else, like signaling or maybe degrading molecules like Cat1 does. You expect a combo.
So you'd think Cat1's NAD chopping ability would be paired up with some other function in the system.
That would be the typical expectation for a type 3 system, yes. But what the researchers found looking at the bacteria that have Cat1 is that in many cases, Cat1 seems to be doing the heavy lifting all on its own.
Really?
It appears to be the main, sometimes maybe the only enzymatic activity responsible for providing the protection in those particular bacteria. It's like a powerful solo artist in a genre usually dominated by duos or groups.
A solo performer. Okay, so let's try and pull this all together. We've got this Cat1 protein. It uses a unique tactic inducing a metabolic freeze by destroying NAD plus Mrugger. It gets triggered by the CA4 alarm signal. Structurally, it's incredibly complex, forming filaments, then trigonal bundles, even pentagonal bundles, and it often seems to work alone, unusually for its system type. What's the big takeaway from discovering all this about Cat1?
Well, as Marafini points out in the discussion, it really drives home just how remarkably effective and incredibly diverse these CRF effectors are. The range of molecular tricks they use to shut down a cell and stop a virus, it's much broader than we might have thought.
They're finding more and more ways bacteria can sabotage an infected cell.
Exactly. And the research clearly shows that Cat1, with its unique approach, works. It's very effective at stopping phage replication and protecting the bacterial community. The big picture, as you said earlier, is confirmed.
I sense a but coming.
But yeah, as the researchers themselves emphasize, while we now know what Cat1 does, the strategy, and what it looks like, the complex structure, there are still big questions about the finer points of how.
Like the why of the bundles.
Precisely. How exactly do those bundles assemble and maybe expand? Is there a specific functional advantage to being a trigonal bundle versus a pentagonal one? How does the NAD plus depletion translate so efficiently into that rapid metabolic freeze? There's still a lot of mechanistic detail to figure out.
So, plenty of intriguing questions left for future research.
Definitely. It opens up whole new avenues.
And, for you, the listener, I think this deep dive into Cat1 is just a fantastic glimpse into the sheer ingenuity of life at the microscopic level.
Yeah.
It's easy to think of bacteria as simple, but then you see something like this.
Yeah, proteins building these elaborate dynamic structures.
Right. It reveals these hidden layers of surprising complexity.
Yeah.
Fundamentally changing how we think about these unseen molecular wars happening constantly, all around us, inside us even.
It certainly gives you a new appreciation for microbes.
Yeah. Absolutely. So thinking about these single-celled organisms, constructing these intricate, expanding spiral protein architectures just to chop up one type of molecule and freeze an infection. Maybe here's a final thought for you to chew on. We often talk about microbial simplicity, but maybe that's just our lack of observation. If a bacterium has evolved molecular machines this sophisticated for defense, what other layers of hidden genius, what unexpected strategies, what complex molecular battles are playing out in the microbial world that we haven't even conceived of yet? Makes you wonder what other surprises are waiting down there at that scale, doesn't it?